Hello. Good evening to the student who have joined. Can you let me know in the chat box if my voice is audible? And also let me know if you can see the video and the presentation. In the chat box, please let me know if everything is fine with the audio, video, and also the presentation. Okay, thank you so much. We will wait till it's 6 p.m. and more students join. Then we will start today's session. Okay, so it's almost 6 p.m. I will start the session. Let me make the call open so that the students who are joining later, they can join easily. So I hope my voice is audible to you and you can see my video also. Let's start today's session. Meanwhile, when more students will join, they will automatically can get in. Okay, let's start with the introduction. So, very good evening to all the students who have joined. My name is Mousumi Deb and I'm the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today, it is our week 9 live session. Previously, we are going ahead with every week. So in our first live session, very first live session, we have covered week 1 and week 2 course content. So subsequently, we are covering the course content which is basically live on that particular week. So right now, the week 10 is live on the SWAYAM portal. And today in our week 9 live session, we are going to discuss the course content which are included in your week 10 basically. Okay. So before starting, I would like to clarify few points which I do before every class. So there will be this kind of problem solving sessions every week. These sessions will be live on Google Meet platform and the timing will be every Monday from 6 p.m. to 8 p.m. for 12 weeks. So we have 12 your course is 12 weeks. So we are going to have 12 classes in on each Monday. And since we are going ahead with one class, so the last Monday we will cover up, summarize the whole course along with one extra class that I will announce later when the extra class will be. In between the class, if any network disruption happen from my side, please wait for some time. Okay, do not leave the session as soon as possible. I will try to join as soon as possible. And if the network issue is from your side, you can rejoin at any time. You don't have to worry about anything. Okay, I am recording the session, so which I will share at the end of the class. Okay, since I am recording the session, I have muted you all and kept your cameras off so that there is no problem in recording the session. But the chat box is always open. So if you have any doubt, any query, you want to tell me anything, you can communicate through the chat box. Okay, your every doubt, every query, anything, you can let me know through the chat box. I will. I'm checking the chat box every now and then. And if as I have already said, if you have any network disruption from your side, you can rejoin at any time because since I'm recording the session, you can check the part you are missing at the later also. And during the session, you can post your doubt and queries. As I already said, that through the chat box, we will communicate. You can let me know your doubts and queries. And if still after the class the doubt persists, you can drop an email to me. I will answer your queries through the email also. This presentation and class recording, it will be shared to you after the class. Okay, so after the class, around after tonight or today morning, you will be able to access the particular course contents. Where do you have to access the course content? In the SWAYAM portal. Okay, I will show you where to access it at the end of the session. Okay. So today, as I have already mentioned, it is our week 9 live session and we are going to cover up the topics which are included in your week 10 course content. So let us first see what are the week 10 course contents. So week 10 course contents are basically very interesting topics are now coming up in today's week also and the upcoming week also. The topics are very interesting and very important for this course because now we are going till now we are what the topic we are covering, it was like biology, some organic chemistry. It was a mix up, but now application portion is coming up, right? So we are going to discuss in today's week and also to the upcoming last 2 weeks. We are going to discuss the application of organic chemistry and medicinal chemistry in the various biological applications. Means in the various drug-related molecules, in various antibacterial, antiviral, anti-cancer drugs. All these things we are going to discuss or starting from today. So today, basically, we are going to discuss about the antimicrobial drugs, the chemistry of penicillin resistant to beta-lactam antibiotics, mechanistic studies of beta-lactamase, non-beta-lactam antibiotics, mechanistic enzymology of isopenicillin N synthesis. Okay. So these are the topics that today we are going to discuss, and all these are basically are in the part of antibacterial drug discoveries. Okay. So let me just check once. So I hope my voice is audible to you, and if you have any doubt or any problem, you let me know always. Okay, the chat box is open. So let me start with the session. So like every week, what we will do is I will show you around 20 questions. All the questions will be MCQs. All the question will have four different options A, B, C, and D. And one of the options obviously will be correct. And after showing a question, I will wait for 2 to 3 minutes. You have to let me know in the chat box A, B, C, or D which of the following options will be correct. And then after some time, I will check the chat box and I will show you the particular question. And then we will discuss that particular question and the topic. In that way, you will have the practice to solve the MCQ type of question, which will help you in solving your weekly assignments. And also, we will discuss the topic in detail. Okay. So let us start with the first question of today's session. So the first question of today's session is, it says, which of the following statements is false for bactericidal agents? Which of the following statements is false regarding bactericidal agents? Your options are: number A, these agents stop the growth of the bacteria but do not kill them. Number B, example includes vancomycin. Option C, the agents destroy the bacteria completely. And number D, the agents do not work with the immune system of the host. So which of the following statements is false for bactericidal agents? Again, I will repeat the options. The first option says these agents stop the growth of the bacteria but do not kill them. Number B, example includes vancomycin. Number C, the agents destroy the bacteria completely. And number D, the agents do not work with the immune system of the host. Which of the following statement is false regarding bactericidal agents? Let me know in the chat box A, B, C, or D which of the following statement is false. Let me know in the chat box. I will wait for 1 or 2 minutes, then I will check it, okay? Okay, let me check the chat box now. Okay, I have two options and both are same, which says the option number A. Okay, let me check if it is correct or not. Yes, option number A is the correct one, means this statement is false regarding the bactericidal agents. Okay, now let us understand what is a bactericid. So there are two terms which are very important in case of this microbial agents. Okay, first is the bacteriostatic, which defines as the antimicrobial agents that reversibly inhibit the growth of the bacteria are called the bacteriostatic. For example, tetracyclines, chloramphenicol. And then bactericidal, those with an irreversible lethal action on the bacteria, is known as bactericidal. For example, penicillin, isoniazid. So these agents have the lethal action on the bacteria, and bacteriostatic, they inhibit the growth of bacteria but do not kill them. So what are the differences between bactericidal and bacteriostatic? Bactericidal refers to the agents that kill the bacteria. Okay, that is why your first statement is false, because it says that the agents stop the growth of the bacteria but do not kill them. This statement is false because bactericidal agents, they kill the bacteria. And bacteriostatic refers to the agent that prevent the growth of the bacteria. The action of bactericidal is irreversible. The action of bacteriostatic is reversible. Bactericidal inhibit the cell wall formation of the bacteria, and bacteriostatic they inhibit the DNA replication and protein synthesis of the bacteria. Bactericidal do not work with the immune system of the host. But bacteriostatic, they work with the immune system of the host to prevent the growth and reproduction of the bacteria. Bactericidal minimal bactericidal concentration or MBC refers to the concentration of the drug required to kill 99.99% of the bacterial population. And bacteriostatic minimal inhibitory concentration or MIC is the minimum drug concentration which inhibits the bacterial growth. For example, the bactericidal examples are the beta-lactam antibiotics, cephalosporin and vancomycin. And for example of the bacteriostatics are the tetracycline, spectinomycin, chloramphenicol and sulfonamide. So why the statement number 1 is false? Now you know that the agents stop growth of the bacteria but do not kill them. It is not bactericidal, it is bacteriostatic. And other statements were right, which says that the example of the bactericidal is the vancomycin, which is correct. The agents destroy the bacteria completely, it is also correct. The agents do not work with the immune system, it is also correct. Okay, so the false statement is number E. So very good, the first question was correct. Now I will go to the second question. The problem number 2 says penicillins exert their antibacterial action by inhibiting cell wall biosynthesis. Which cell wall biosynthetic enzyme is inhibited by penicillin? Your options are number A, carboxypeptidase, number B, transpeptidase, number C, glycosylase, and number D, phosphatase. Which of the following enzyme is inhibited by penicillin? Penicillins exert their antibacterial action by inhibiting the cell wall biosynthesis. Which of the following enzyme is inhibited? Number A, carboxypeptidase, number B, transpeptidase, number C, glycolysis, and number D, phosphatase. Again, I will give you some time. Let me know in the chat box, A, B, C, or D, which one of the following is the correct one. Okay, let me check the chat box now. Okay, number B and number A. So I have two different options for this question. Let me check which of the following is the correct one. So the correct answer is, it is number B. Transpeptidase is the correct answer. Transpeptidase is the enzyme which is inhibited by the penicillins. So what is the mechanism of action? The cell wall biosynthesis inhibition is a mechanism of action of the penicillin. So penicillin inhibits the final cross-linking stage of the cell wall synthesis. It reacts with the transpeptidase enzyme to form an irreversible covalent bond. Inhibition of transpeptidase leads to a weakened cell wall. Cells swell due to the water entering the cell and then burst, which is known as lysis. So lysis is the disintegration of the cell by the rupture of the cell wall or membrane, and this lysis is mediated by the penicillins. The penicillin possibly act as an analog of L-ala gamma D-ala portion of the pentapeptide chain in the transpeptidase enzyme. So this is the structure of penicillin, as you can see here, and this is the structure of acyl D-ala D-ala. This is the pentapeptide, okay, and this pentapeptide is mimicked by the penicillin, and that's how they act as a interacts with the transpeptidase enzyme, and then they inhibit the cell wall biosynthesis. So what is the beta-lactam mechanism of action, as I have already mentioned? So I will discuss it later also. So it mimics the structure DLR, it structurally mimics this particular pentapeptide, okay, and that is how it ruptures the cross-linking between this particular cell membrane, and this due to the interruption of the cross-linking and cell wall synthesis, the cells swell and they burst. So the method is the lysis also. So the transpeptidase was the correct answer. Now I will go to the next problem. Problem number 3 says the three simple building blocks of penicillins are, your options are number A glycine, cysteine, valine, number B alpha amino adipic acid, cysteine and leucine, number C beta amino adipic acid, cysteine and isoleucine, number D alpha amino adipic acid, cysteine and valine. Which of the following is the correct one? The three simple building blocks of the penicillin are number A glycine, cysteine, valine, number B alpha amino adipic acid, cysteine and leucine, number C beta amino adipic acid, cysteine and isoleucine, and number D alpha amino adipic acid, cysteine and valine. Which of the following statement is the correct one? What are the three simple building blocks of penicillin? Let me know in the chat box. Let me check the chat box now. Okay, I have two answers. Both our answers are saying it is the number D. Let me check if number D is the correct one or not. So the correct answer is very good. It is number D, alpha amino adipic acid, cysteine and valine. These are the three simple building blocks of penicillin. So this is the structure of penicillin. So remembering the structure of penicillin is very important. So as you can see, the structure of the penicillin, we have a four-member ring, which is the beta-lactam ring. Okay, so there are the names. So lactone means the cyclic esters. Okay, and the lactams are the cyclic amides. So beta-lactam says there are two carbon here, alpha carbon and beta carbons. That is why it is known as a beta-lactam ring, and it is fused with a five-member ring which has a sulfur, and they there are specific stereochemistry, and in some position, remembering the stereochemistries are also very important. Okay, because this stereochemistries also influence the biological activity of penicillins. And this R group is variable. So penicillin, there are different penicillins present, which is because of the variation of the R group. So there are three simple building blocks from where this penicillin can be derived. So first is the alpha amino or L-amino adipic acid. Then we have L-cysteine and D-valine. Okay, and the combination of these L-amino adipic acid, L-cysteine, D-valine forms the ACV tripeptide. So this is the structure of ACV tripeptide. Then this ACV tripeptide, it forms the isopenicillin N. Okay, and this isopenicillin N forms the penicillin. Ultimately, it forms the penicillin and also penicillin G and also the cephalosporins. So this is the form called transformation, and I suggest to remember this transformation. So how this alpha amino adipic acid, L-cysteine and D-valine forms the penicillin. Try to remember this transformation. It will be important. Okay. Now I will go to the next problem. So problem number 4 says, which reaction is catalyzed by a beta-lactamase enzyme? Your options are: number A, the final cross-linking reaction to form the bacterial cell wall. Number B, the hydrolysis of the acyl side chain of penicillin. Number C, the hydrolysis of the four-member ring in penicillin. Number D, the biosynthesis of penicillin from LLDACV. So which of the following statement is correct? Which of the following reaction is catalyzed by beta-lactamase enzyme? Again, I will repeat. Option A, the final cross-linking reaction to form the bacterial cell wall. Number B, hydrolysis of the acyl side chain of penicillin. Number C, hydrolysis of the four-member ring in penicillin. Number D, the beta biosynthesis of the penicillin from LLD-ACV. Which reaction is catalyzed by beta-lactamase enzyme? Okay, let me check the chat box. Okay, I have only one answer, which says it is number C. Others also try. I will wait for 1 more minute. Okay. Again, I will repeat. Which reaction is catalyzed by beta-lactamase enzyme? Your options are: number A, the final cross-linking reaction to form bacterial cell wall. Number B, the hydrolysis of the acyl side chain of penicillin. Number C, the hydrolysis of the four-membered ring in penicillin. Number D, the biosynthesis of penicillin from LLDACV. Which of the following statement is correct? Again, let me check the chat box if there is any another answers. Okay, I have now another answer which says it is number C. Okay, let me check. Okay, there is a power cut. Hello, am I audible? So there is a power cut, that is why there is a problem. Hello, let me know if I am audible to you. Okay, thank you for confirming. Okay. Okay, so let me show you what is the correct answer. So the correct answer is number C. The hydrolysis of the four-membered ring in penicillin. This reaction is catalyzed by the beta-lactamase enzyme. Okay. So this is the beta-lactam ring. Now I have already mentioned you lactam. This term means cyclic amides. Okay, so cyclic amides are known as the lactam, and your cyclic ketones are known as the lactones. So this is known as beta-lactam ring. Why this is known as a beta-lactam ring? Because here we have two carbons. So this carbon which is next to the carbonyl group, it is known as the alpha carbon, and the carbon next to it, it is the beta carbon. Since we have two alpha and beta carbons here, so this particular ring is known as the beta-lactam ring. So now what is the role of the beta-lactamase enzyme? So as the name suggests, lactamase means it will hydrolysis the particular beta-lactam ring. So as you can see, this is the structure of the penicillin. Okay, and this in the penicillin, I have mentioned you already, there is a beta-lactam ring, and beta-lactamase enzyme, what does it do? It hydrolyzes this particular lactam ring. And now what is the what forms from the particular hydrolysis of the amide, which will be carboxylic acid and the amine. Okay, so as you can see, when this ring breaks, this kind of formation, carboxylic acid and amine is forming here. So there are two type of beta-lactamase mainly. One is the serine beta-lactamase and another is metallo-beta-lactamase. So in the serine beta-lactamase, what happened? This is the serine beta-lactamase, the active site of serine beta-lactamase. Here we have three important amino acid. One is glutamine 166, one is lysine 73, and one is serine. So what does this serine do? This serine acts as a nucleophile. There is a water molecule in the active site of the particular enzyme. This serine molecule amino acid has this OH group which act as a nucleophile and attacks to the carbonyl carbon of the beta lactam ring, and this ring breaks. Okay, and this glutamate it helps to deprotonate this nearby water molecule to form hydroxyl ion, which is more powerful or more potent nucleophile. It will again attack to the carbonyl carbon, and then this we will get this particular enzyme and we will have this carboxylic acid. With the first step is the acylation step, followed by the second step, which is the deacylation step. And also we have the metallo-beta-lactamase. Now what is the metallo-beta-lactamase? So metallo-beta-lactamase has the metal ion which is the zinc ion in the active site, and this there is a zinc bound OH group. This zinc bound OH group act as a nucleophile and attacks to the particular carbonyl carbon of the ring, and that's how this particular mechanism of action shows. Okay, so the correct answer was the hydrolysis of the 4-member ring in penicillin is the correct one. Okay, and this is the reaction which is catalyzed by the beta lactamase enzyme. Okay, so yeah, number C was the correct one. Okay, so I think you have understood it. If there is any problem, let me know. Okay, now I will go to the next problem. So problem number 5 says, find out the incorrect statement among the following. Your options are: number A, moxifloxacin is a quinolone class of antibiotic drug. Number B, method of action of quinolones is the inhibition of the DNA guidase. Number C, doxycycline works by stopping the DNA replication. Number D, tetracyclines prevent the binding of tRNA with A site. So which of the following statement is incorrect? Read all the options very carefully because the statements have different it is regarding different classes of antibiotics. So identifying the correct answer is very important. So which of the following statement is incorrect? First option, moxifloxacin is a quinolone class of antibiotic drug. Number B, method of action of quinolones is the inhibition of DNA guidase. Number C, doxycycline works by stopping the DNA replication. Number D, tetracyclines prevent the binding of tRNA with A site. Find out the incorrect statement among the following. Moxifloxacin is a quinolone class of antibiotic drug. Number B, method of action of quinolones is by inhibition of DNA guidase. Number C, doxycycline works by stopping DNA replication. Number D, tetracyclines prevent the binding of tRNA with the A site. Let me check the chat box. Okay, I have one answer which says it's number C. Okay, 1 second. Just connect. Hello, am I audible? Kindly let me know if I am audible or not because there is some disruption. Okay, fine, thank you. Okay, let me show the correct answer. So I think I have only one answer in the chat box which says it's number C. So let me check if number C is the correct answer or not. Please bear with me because there are some network disruption today due to the poor weather. There are frequent power cuts, so please bear with me. So let us see what is the correct answer of problem number 5. So the correct answer of problem number 5 is, it is number C. Doxycycline works by stopping DNA replication. This statement is incorrect. So now let us understand why this statement is incorrect. So this is the structure of the doxycycline. So as the name suggests, cycline means it has a fused cyclic structure. Okay, so there are several cyclic rings which are fused with each other in the doxycycline. So what is the mechanism of action? So doxycycline is a part of the tetracycline class of antibiotics. So tetracycline means there will be four different rings which are fused together. That's the name suggests as tetracycline. So tetracycline is a specific inhibitor of bacterial protein synthesis. They bind to the 330S ribosomal subunit and thereby prevent the binding of the amino acid tRNA to the mRNA ribosome complex. Tetracyclines also inhibit the protein synthesis in the host, but are less likely to reach the concentration required because eukaryotic cells do not have a tetracycline uptake mechanism. Okay, so this is the structure. This is the amino acid tRNA which binds to the particular A site of the mRNA ribosome complex. But when the tetracyclines binds to this A site, the amino acid tRNA cannot bind to the particular site, and then the protein synthesis it is disrupted. Okay. So now let us recapitulate all the statements which we are present in this question. So the first statement says moxifloxacin is a quinolone class of antibiotic, which is correct. Okay. Moxifloxacin is an antibiotic which is in the quinolone class. Method of action of quinolones is the inhibition of DNA kinase. Is it also correct? Doxycycline works by stopping DNA replication. It is false because doxycyclines are a part of tetracycline class of antibiotic, which mechanism of action are the prevention of the tRNA with the A site, and thus it prevents the protein synthesis. Okay, so one thing you have to do is remembering different class of antibiotics along with their examples. So sometimes the question can come indirectly, like you are not given the particular class, you may be given the particular name of the drug. For example, instead of given the mechanism of action of tetracycline class of antibiotics, the question can be given that write the mechanism of action of doxycyclines, and so on. Okay, so remembering a particular what is the things is very important. The things are very important are remembering particular class of antibiotic, their examples, their structures along with their mechanism of action. These things you have to remember. Okay, even if you do not thoroughly remember the structure, but just remember how it looks in overall. Like for doxycycline, for tetracyclines, remember there are 4 cyclic rings. In the penicillin, in the beta lactamase inhibitor, so it has a beta penicillin beta lactam ring, and so on. Okay, so these things are remembering is very important. So now let me go to the next problem. Problem number 6 says augmentin is a life-saving drug, and it is a mixture of, your options are number A, oxacillin and ampicillin. Number B, amoxicillin and clovinic acid. Number C, oxacillin and clovinic acid. Number D, amoxicillin and pyruvic acid. Augmentin, a life-saving drug, is a mixture of number A, oxacillin and ampicillin. Number B, ampicillin and clovinic acid. Number D, oxacillin and clovinic acid. And number D, ampicillin and pyruvic acid. So which of the following statement is correct regarding the augmentin? Again, all of the students present in the class try to answer the questions. Okay, if you try to answer the question, even if your answer is wrong, it's fine, because I'm not giving you any marks regarding to the answering today. But answering is important for yourself, because it will you will understand that you are learning the course better or not. Okay, and also answering here, it will help you in answering your weekly assignments also, which have the marks, right? So try to answer the question. Augmenting a life-saving drug is a mixture of which two components? Let me check the chat box. Okay, I have again one answer. Others are not answering, but I want all of you to answer. Okay. So B is the given answer. Let me check if B is the correct one or not. Okay, so the correct answer is very good. It is B, amoxicillin and clavonic acid. So augmentin. So these are the structures of amoxicillin, and this is the clavonic acid. So amoxicillin is a type of penicillin. Okay, where the side chain is this. And this is the structure of the clavonic acid, which also have its beta-lactam ring along with the other things. Okay, so this is the augmentin. So this is the so there are two part of this augmentin. One is for amoxicillin, which is because which is responsible for the bacterial cell death, and which is the beta-lactamase. Okay, and the ring open cleavage product of the amoxicillin. So the ring open we can product of the amoxicillin refers to the beta-lactamase, and potassium clavulate, it is the inhibition of the particular enzyme. Okay, so this is the two roles of this particular amoxicillin and potassium clavulate, and the augmentin has these two components. Okay. So problem number 7 says erythromycin belongs to dash class of antibiotics. Your options are number A macrolide, number B tetracycline, number C oxalidione, number D quinolone. So erythromycin belongs to which class of antibiotics? Macrolide, tetracyclines. Okay, the this if spelling is incorrect. Okay, the tetracycline it will be then oxalidione and then quinolone. Erythromycin belongs to which class of antibiotics? Macrolide, tetracycline, oxalidione, quinolone. Number B is the tetracycline. There is a spelling mistake which I will correct soon. All the students who are present, please respond, okay, and let me know what is the answer you think is the correct one. Even if your answer is wrong, it's fine, but answering is very important, okay? Erythromycin belongs to which class of antibiotics? Let me check the chat box. Okay, one answer I got, which is says it's number A. Let me check if it is correct or not. So the correct answer is yes, it is number A. It is the macrolide. Okay, first let me correct it out. It will be tetracycline. So it is the macrolide class of antibiotic, erythromycin. So it is macrolide means it will be a cyclic compound, but it has it is a larger number of the bonds and the carbons will be there, which is the macrolide, which is means this will be extra large ring of the compound. So the mechanism of action, erythromycin displays the bactericidal activity, particularly at higher concentration. It prevents the growth of the bacteria by inhibiting their protein synthesis. Erythromycin binds to the 23S rRNA molecule in the 50S ribosomal subunit. Okay, this binding blocks the three exit from the blocks the exit of the growing peptide, thus inhibiting the translocation of the peptides. Okay. So, erythromycin inhibits the translocation. It binds to the 20S rRNA molecule in the 50S subunit. Here, it binds. So, there are two sides. One is A side, one is P side. I have mentioned that the tetracyclines, they binds to the A side. Okay, and the erythromycin binds in between the A and P side. So, what does it do? It blocks the exit of the growing peptide chain by inhibiting the translocation, and that's how it inhibits the protein synthesis in the bacteria, exerting its antibacterial effect. So now there are different type generations of the macrolide. In the first generation is the erythromycin A, which has this structure, which has a six-membered macrolide ring in along with the deoxy sugar. Then we have second generation is the azithromycin, which is very very famous antibiotics and very vastly used in nowadays is the azithromycin, which has this 15-membered macrolide ring, okay, along with the nitrogen in the ring portion and the deoxy sugar. And then third generation we have erythromycin, which has a 14-membered ketolide ring along with the deoxy sugar. Okay, so these are the different generation of macrolides. You do not need to remember the structures here, but just remember how it looks, because the question can be given like the structure is given and you are asked the question regarding to the structure. Okay, so it can happen. That is why I am telling you to remember the structures. Okay, and the mechanism of action, obviously to remember it, so it binds to 20th RNA molecule and inhibits the protein synthesis. Now problem number 8, it says IPN to 6-APA conversion is done by number A, ACVS, number B, IPNS, number C, TAOCS, and number D, IPNA. IPN to 6-APA conversion is done by which enzyme? ACVS, IPNS, DAOCS, and IPNA. Which of the following will be the correct answer? IPN to 6-APA conversion is done by what? So all these are the enzymes, okay, and they are the abbreviation forms I have given, because sometimes so many times you have seen that the abbreviations are given for the enzyme. So remembering the abbreviation of the enzymes are very important. So IPN to 6-APA conversion is done by which enzyme? Let me check the chat box. I have one answer. It says number B. Let me check if number B is the correct one or not. So the correct answer is, it is number D, IPNA. So IPNA is the enzyme which is responsible for the conversion of IPN to 6-APA. Now let me show you the conversions. Okay, so this question I have already given you earlier also. The building blocks of particular penicillin are the alpha amino adipic acid, L-lysine and L-valine. Okay, or D-valine also you can say. So alpha amino adipic acid, cysteine and valine, they combine to form a AC combine to form delta L alpha amino adipoyl L cysteine D-valine, which is the ACV. Okay, and the enzyme which is responsible for it is the ACV synthetase or ACVS. Okay. Then this ACV is converted to the IPN by IPNS, which is the IPN synthesis, means isopenicillin N synthetase. Okay. Then this isopenicillin N synthesis N is converted to 6-amino penicillinic acid, okay, or 6-APA by the IPN amino hydrolase or acyl transferase, which is IPNA. And then this 6-amino penicillinic acid or 6-APA is converted to other penicillins by IPN amino hydrolase or acyl transferase, this same enzyme. Okay. Then isopenicillin N can also be converted to penicillin N by IPNE or IPN epimerase, and penicillin N is converted to D-acetoxycephalosporin or C or DAOC by DOACS or DOAC synthetase or DOAC DAC synthetase. Okay, and this DOAC or D-acetoxycephalosporin C is converted to other cephalosporin and cephamycins by other routes. Okay. So that is why I have told you to remember all the abbreviation also. So IPN means isopenicillin N converted to 6-APA means 6-amino penicillin 6-amino penicillin acid. Okay, and the enzyme responsible is IPNA or IPN amino hydrolase or IPN acyl transferase. Okay, IPNS works here when the ACV is converted to IPN. Okay, then the IPN or S or IPN synthetase, this enzyme get activated, and isopenicillin or IPN is converted to penicillin N by IPN epimerase, which is followed by the DAOC synthetase to convert it to the D-acetoxycephalosporin C. So remembering these conversions are very important. So I suggest you to remember this scheme and all the conversions, because this is very important for your this topics. Okay. Now I will go to the next problem. Problem number 9 says, which metal is present in the active site of isopenicillin N synthetase? Your options are number A magnesium, number B iron, number C cobalt, and number D nickel. So which metal is present in the active site of isopenicillin N synthetase? Which of the following metal is present in the active site of isopenicillin N synthase? Magnesium, iron, cobalt, and nickel. Which metal is present in the active site of isopenicillin N synthase or IPNS? Let me check the chat box now. Number B is the given answer. Others are not answering. I want all of you to answer, okay? Okay, let me check if number B is the correct one or not. So the metal which is present in the active site of IPNS is That is the iron, which is number B, which is the correct answer. Okay. So this is the IPNS ferrous active site in absence of the substrate. So we have a iron. With this iron, there are the coordination of the histidine amino acids, and then we have the glycine, okay, and then aspartic acid and water, okay. And then we have this ACV. So IPNS, as you remember, it works on the ACV to convert ACV to isopenicillin N. And when the ACV is there, it attacks here and binds to through its sulfur side. Okay, and it is the IPNS ferrous active site with ACV bound. Okay, you don't need to remember the active site. Okay, but remembering the iron metal ion is important because sometimes the question can be asked regarding this. But active site, if you can remember, it is fine. If you cannot remember it, it is also fine. So iron is the correct answer. This is the metal ion which is present in the active site of IPNS. Now I will go to the next problem, problem number 10, which says that D-cycloserine exerts its antibacterial action by inhibiting number A, L-alanine racemase, number B, carboxypeptidase, number C, carbonic anhydrase, and number D, L-serine racemase. So D-cycloserine exerts its antibacterial action by inhibiting which of the following enzymes? Your options are number A, L-alanine racemase, number B, carboxypeptidase, number C, carbonic anhydrase, and number D, L-serine racemase. D-cycloserine exerts its antibacterial action by inhibiting which enzyme? So D-cycloserine exerts its antibacterial action by inhibiting which enzyme? Okay, let me check the chat box. I have one answer which says it's number A. Okay, let me check if number A is the correct answer or not. So the correct answer is, it is number A, L-alanine racemase is the enzyme which is inhibited by the D-cycloserine. So cycloserine is an antibiotic produced by the Streptomyces orchideus. Okay, mechanism of action, it inhibits the incorporation of D-alanine into the peptidoglycan pentapeptide by inhibiting alanine racemase, which converts L-alanine to D-alanine and D-alanine and D-alanine ligase, finally inhibiting mycobacterial cell wall synthesis. Cycloserine used exclusively to treat tuberculosis caused by the Mycobacterium tuberculosis resistant to first line agents. So this is the chart which is very important for your antibacterial agents. Okay, so it is the bacterial cell, and there are different targets which different antibacterial agents are worked. Okay, so first is the cell wall, which is this one. Okay, the cell wall is inhibited by the cell wall synthesis inhibited by the D-cycloserine, vancomycin, bacteriotricin, penicillin, cephalosporins, and cyclamycins. And cell wall integrity, it is particularly inhibited by the beta-lactamases. And then we have the DNA. Here it's a DNA synthesis, the replication. It can be DNA synthesis is inhibited by the metronidazole, and DNA guidase by the quinolone class of antibiotic. Then RNA polymerase can also be inhibited, and in RNA polymerase by rifampicin. Okay, and then cycloplasmic membrane. Cycloplasmic membrane is the phospholipid membrane by polymyxis. Then translation protein synthesis there are two types of protein synthesis inhibitors. One is the 50S inhibitors, which are the erythromycin, chloramphenicol, syndamycin, and lincomycin, and protein synthesis 30S inhibitors are the tetracycline, streptomycin, and kanamycin. And then so as you can remember, so there are the ribosomes. So this chart, remembering this chart works very well because here there are pictures of different targets and the name along with that the name and mechanism of action of different antibiotics. So I will suggest you to remember this diagram very carefully because it will be very helpful for your antibacterial agents. Okay. Now I will go to the next problem. Problem number 11 says identify the incorrect statement regarding the SAR of penicillin. SAR I have discussed in the last class means the structure activity relationship of the penicillin. The options are number A, thiazolidine ring increases the beta lactam ring strain. Number B, carboxylic group is not essential. Number C, variations are limited to R groups only. And number D, only amide is not involved in binding. So which of the statement is incorrect regarding the ACR of penicillin? Number 8, thiazolidine ring. This ring increases the beta lactam ring strain. Number B, carboxylic acid group. This group is not in essential. Number C, variation are limited to this R group only. Number D, only amide. This one is not involved in binding. So which of the following statement is incorrect regarding the ACR of penicillin? Read all the options very carefully, then answer it. And I want all of you to answer this question. The students who are present in the class, please answer the question. Identify the incorrect statement regarding SAR of penicillin. Let me check the chat box. Okay, number B is given answer. Okay, I think I have number B as the answers. Okay. So the correct answer is yes, it is number B. Carboxylic acid group is not essential. This statement is incorrect. So these are the different your SAR of the penicillin basically. So the first is the amide and carboxylic acid are involved in binding. Okay. This carboxylic acid binds as a carboxylate ion. The mechanism of action involves the beta-lactam ring. Activity related to the beta-lactam ring strain, which is subject to stability factors. Bicyclic system increases the beta-lactam ring strain. Not much variation of the structure is possible. Variations are limited only to the side chain. So here, as you can see the structure, this amide is essential, this beta-lactam is essential also, this bicyclic system is essential, carboxylic acid is essential, and one thing is essential is the cis stereochemistry is in here, two hydrogens is stereochemistry is essential. Okay, and this mechanism of action involves the beta-lactam ring. Bicyclic ring, because of this thiazide ring, increases the strain in the beta-lactam structure, and the variations are only limited to the particular amide side chain. Okay, and the amide and carboxylic acid, these groups are involved in binding. So this is the SAR or the structure activity relationship of the penicillin, which is very important for you to remember. Okay. Now I will go to the next problem. Problem number 12 says clavonic acid is, your options are number A, an irreversible inhibitor of the transpeptidase enzyme. Number B, a reversible inhibitor of L-alanine racemase. Number C, a suicide inhibitor of serine-based beta-lactam inhibition enzyme. Number D, a reversible inhibitor of serine-based beta-lactamase enzyme. Which of the following is the correct answer? Clavonic acid is number A, an irreversible inhibitor of transpeptidase enzyme. Number B, a reversible inhibitor of L-alanine racemase. Number C, a suicide inhibitor of serine-based beta-lactamase enzyme. And number D, a reversible inhibitor of serine-based beta-lactamase enzyme. Which of the following is the correct answer? Clavulanic acid is which type of inhibitor? Is it a reversible inhibitor of the transpeptidase enzyme? Or is it a reversible inhibitor of L-alanine racemase? Or a suicide inhibitor of serine-based beta-lactamase enzyme? Or a reversible inhibitor of serine-based beta-lactamase enzyme? Let me know which of the following is the correct statement. Let me check the chat box. Okay, I have one answer which says it's number C. Let me check if number C is the correct answer or not. So the correct answer is very good. It is number C. It is a suicide inhibitor of the serine-based beta-lactamase enzyme. So this is the structure of the clavulanic acid. As you can see, we have a beta-lactam ring here followed by a fused ring system which has a carboxylic acid group here and a double bond followed by a alcoholic group. So the clavulanic acid widely stays as the clavulanic potassium clavunate. This is a systematic RBC name which you do not need to remember. Okay, clavulanic acid can be considered as the most important and representative among the inhibitors of the beta lactamases. It is the first clinically useful beta lactamase inhibitor. Was identified as a natural product from the strain of the Streptomyces clavineus. So structurally, it is one oxapenum lacking in 6-acyl amino acid of the penicillin but possessing the 2-hydroxyethylene dioxy moiety as the C2. So this moiety, which is known as the your 2-hydroxyethylene diene moiety. Okay, so here we don't have a amide side chain, but we have this ethylene the moiety in the C2 position. So potassium, so this is the, as I have already mentioned about the augmentin, which is the combination of the amoxicillin and the potassium clavonate. Okay, so penicillinase enzyme or beta lactamase attacks to this particular the penicillin. Okay, and then from penicillin it follows the penicillinic acid, which is antibacterially inactive. Okay, so what does this particular potassium clavonate do? So it potassium clavonate, it inhibits the action of the penicillinase enzyme or the beta lactamase enzyme. So now understand, let us understand about the augmentin. I haven't mentioned it in detail in the last question because I was waiting for this question. So basically, what is the reason to use two different combination of the drugs in the augmentin? We are using amoxicillin, which is an antibacterial drug. It is fine, okay, because it is the antibacterial drug which will inhibit the cell wall synthesis and so on. But what is the reason of using this clavulanic acid or the potassium clavonate? Because we have the beta lactamase enzymes, and this beta lactamase enzyme, what does, or also they are known as the penicillinase enzyme. What does they do? Now, they basically attacks to the beta lactam ring and breaks this beta lactam ring, followed which and makes it the penicilloic acid, which is not antibacterially active. It is inactive. Okay. So potassium clavonate, what does it do? It inhibits this beta lactamase enzyme and attacks it, and it is in case of the penicillin resistant bacteria, this enzyme is attacked by the potassium clavonate and by forming a suicide substrate, and that's how now this particular mixed amoxicillin cannot be hampered by this beta lactamase enzymes and it can exert its antibacterial activity. That is why combination of these drugs in the potassium clavonate and amoxicillin is very important, because for the resistant bacteria, because this is the logic behind the formulation of the augmentin. Okay, so remember, clavulanic acid is a very important portion, and it is the suicide inhibitor of the penicillinase or beta lactamase serine beta lactamase enzyme. Okay, and this is the structure of clavulanic acid. The structure remembering is also very important, so remember the structure. So now I will go to the next problem. Problem number 13 says, which of the following statement is false regarding gram-positive bacteria? Your options are number A, these are very much susceptible to antibiotics. Number B, they have low lipid content. Number C, peptidoglycan layer is thin, majorly single layer. Number D, outer membrane is not present. So which of the following statement is false regarding gram-positive bacteria? Your options are number A, they are very much susceptible to antibiotics. Number B, they have low lipid content. Number C, peptidoglycan layer is thin, majorly single layer. Number D, outer membrane is not present. So which of the following statements regarding the gram-positive bacteria is false? I will give you 2 minutes, then I will check the chat box and I will discuss it, okay? Let me check the chat box now. Okay, I have one answer which is says it's number D. Let me check if number D is the correct answer or not. So, the statement which is false regarding the gram-positive bacteria is, it is number C. Peptidoglycan layer is thin, majorly single layer. This statement is false. So differences between the gram-positive and gram-negative bacteria. First is the cell wall. So cell wall is single layered, smooth in case of gram-positive, and it is double layered and wavy in case of the gram-negative bacteria. Cell wall thickness. The thickness of cell wall is 20 to 80 nanometers in the gram-positive, and in the cell wall thickness is 8 to 10 nanometer in the gram-negative bacteria. Peptidoglycan layer is thick and can be multi-layered in the gram-positive, and thin and single layer in case of the gram-negative. That is why this statement is false, which says that peptidoglycan layer is thin and majorly single layer. This is false regarding the gram-positive, because in gram-positive the peptidoglycan layer is thick and it can be multi-layered, whereas in the gram-negative bacteria it is thin and often single layered. The teichoic acids are present in case of gram-positive bacteria, but these are not present in gram-negative bacteria. Lipopolysaccharide is not present in gram-positive bacteria. Lipopolysaccharide is present in gram-negative bacteria. The outer membrane is not present in gram-positive bacteria. The outer membrane is mostly present in the gram-negative bacteria. Lipid content is very low in case of gram positive, but lipid content is about 20 to 30% in case of the gram negative bacteria. Resistance to antibiotic, these are very susceptible to antibiotics, but the gram negative bacteria are very resistant to antibiotics. So these are the differences between the gram positive and gram negative bacteria. So the statements, the number A, it says the gram positive bacteria is susceptible to antibiotics, which is correct. The second statement says it has low lipid content, which is correct. Number C is false, which says the peptidoglycan layer is thin and single layer, whereas it is originally thick and multi-layered. And outer membrane is not present, which is also correct, because in case of the gram positive bacteria the outer membrane is not present, whereas in case of the gram negative bacteria the outer membrane are mostly present. Okay, so the correct answer was number C. So remember the differences between the gram positive and gram negative bacteria along with the examples. I haven't mentioned the examples here. You will find it in any book, or you can Google it also, the examples of gram positive and gram negative bacteria, which is also very important. Okay. Now I will go to the next problem. Problem number 14 says, which of the following antibiotics works by targeting the DNA guidase? Your options are number A, sulfonamide, number B, tetracycline, number C, quinolones, and number D, glycopeptides. Which of the following antibiotics works by targeting the DNA guidase? Sulfonamide, tetracycline, quinolones, and glycopeptides. I will wait for 2 minutes, then I will show the answer. All of you try to answer this question. Which of the following antibiotics works by targeting DNA guidase? Sulfonamide, tetracycline, quinolones, glycopeptides. Which of the following antibiotics works by targeting DNA guidance? Let me check the chat box now. Okay, I have one answer which says number C. Okay, let me check if it is correct or not. So the correct answer is yes, it is number C, quinolones. So quinolones work by targeting the DNA guidance. Okay. So I have mentioned it before also that there are different antibiotics and along with their mechanism of action. So majorly there are three mechanisms through which the antibacterials work. First is the cell wall synthesis inhibition. There are two class of antibiotics like beta-lactams like penicillin, cephalosporin, carbapenems and monobactams, vancomycin, bactricin and also cell membrane the polymaxins. Okay, so these inhibit the cell wall synthesis. The protein synthesis are inhibited by the 50th subunit inhibitors and 30th inhibitor. The 30th subunit inhibitor are the tetracyclines and aminoglycosides. 50th subunit inhibitors are the macrolide, clindamycin, linezolide, chloramphenicol, and streptogrammancin. Then we have the nucleic acid synthesis inhibitors. We have the folate synthesis inhibitor, the sulfonamides and trimethoprim. The sulfonamide inhibits the para-aminobenzoic acid to DHFA conversion, and DHFA to THFA conversion is inhibited by the trimethoprim. Then we have the DNA guidance inhibitors, which are the quinolones, which are we are going to discuss now. They are RNA polymerase inhibitors are the rifampicin. Okay, so these are the nucleic acid synthesis inhibitors. So DNA guidase is an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of the double-stranded closed circular DNA. Guidase belongs to a class of enzyme known as the topoisomerase that are involved in the topological transitions of the DNA. Topoisomerases are able to change the DNA topology to facilitate the function of the such as DNA replication and transcription. Okay, so this is the topoisomerase enzyme, and here the fluoroquinolones or quinolones bind to inhibit this particular enzyme. Okay, so this is the structure of the quinolone. So we have here also the structure remembering is very important. So quinolone binds to the DNA guidase complex blocking the further DNA replication. Okay, so these quinolones are bactericidal. They inhibit the bacterial DNA synthesis in several ways, causing the rapid cell death. Okay, so this is the structure of quinolone. As you can see, this portion, the upper portion is responsible for the binding to DNA. This lower portion is responsible for the binding to the stacking domain. Here is the binding to the enzyme, and this portion also binds to the enzyme. So the sides portion are binds to the enzyme. The upper portion here, when the ketone groups are there, which binds to the DNA, and the lower portion it is the binds to the stacking domain. Okay, so yeah, quinolone is the correct one, and also I haven't given here examples, but remember all the read about the examples of the quinolone class of the compounds. Okay, like moxifloxacins, these are the quinolone class of compounds. Okay. Now I will go to the next problem. Problem number 15 says the minor product formed by acidic degradation of the penicillin G is, your options are number A phenol, number B penicillinic acid, number C phenyl acetic acid, and number D phenyl acetylene. So the minor product formed by the acidic degradation of penicillin G is phenol, penicillinic acid, phenyl acetic acid, and phenyl acetylene. The minor product formed by the acidic degradation of penicillin G is number A phenol, number B penicillinic acid, number C phenyl acetic acid, number D phenyl acetylene. Let me check the chat box. Okay, I have one answer which says number C. Let me check if it is the correct one or not. Yes, number C is the correct one, which says that phenylacetic acid. So this is the structure of the penicillin G. Okay, and the penicillin G acylase or PGA. This enzyme degrades this particular penicillin G to form the 6-APA or 6-aminopenicillinic acid, which has this structure, and phenylacetic acid, which has this structure. So basically, this enzyme is doing, it is breaking this particular amide linkage, forming amine portion, which is the 6-aminopenicillinic acid, and then phenylacetic acid, which is the acidic portion, now which has this structure. Okay, so yeah, phenylacetic acid is the correct answer. Now problem number 16 says the transformation of penicillin sulfoxides to cephalosporins is termed as number A, Pomeroy rearrangement, number B, Morin rearrangement, number C, Smith rearrangement, number D, Claisen rearrangement. Again, I will repeat, transformation of penicillin sulfoxide to cephalosporins is termed as number A, Pomeroy rearrangement, number B, Morin rearrangement, number C, Smith rearrangement, and number D, Claisen rearrangement. Which of the following is the correct answer of problem number 16? Let me know. So the transformation of penicillin sulfoxide to cephalosporins is termed as which type of rearrangement? Is it Pomeroy rearrangement? Is it Morin rearrangement? Is it Smith rearrangement? Or is it Claisen rearrangement? Let me check the chat box. Okay, I have one answer which says it is number A. Let me check if number A is the correct answer or not. So the correct answer is, it is number B, Morin rearrangement. Sulfonic acids or RSOH are among the most common sulfur-centered reactive intermediates generated in biological systems. Okay, so given the biological occurrence of the sulfonic acids, it is important to explore the reactivity of these intermediates under physiological conditions. The Morin rearrangement is a synthetic process developed for the conversion of penicillin derivatives into cephalosporins that proceeds via nucleophilic attack of an alkene on a sulfonic acid intermediate. In its classic form, the Morin reaction involves the initial elimination of the sulfonic acid form cyclist sulfoxide followed by the intramolecular cyclization of the resulting alkene and the sulfonic acid groups to generate an episulfonium ion intermediate that undergoes further reaction to yield the ring expanded products. On the basis of the existing literature, it is difficult to assess whether the reaction between the alkene and the sulfanic group can occur under mild condition, because the conditions required to generate the sulfonic acid from the sulfoxide precursor in the Moring reaction typically involve high temperatures and strong acid. In the work described here, beta-lactam beta sulfonyl ketone precursors were used to generate a Moring type sulfonic acid intermediate under the mild condition. This approach made this possible to demonstrate the intermolecular cyclization of the alkene to phenyl sulfonic acid to generate episulfonium intermediate that can occur when neutral sulfonation in the room temperature. Okay. So remember the name, the Moring rearrangement, which is involved in the transformation of the penicillin sulfoxide to cephalosporins. Now I will go to the next problem. Problem number 17 says, which of the following class of penicillins is synthetically formed, acid stable, and can be taken orally? Your options are number A penicillin G, number B penicillin V, number C ampicillin, and number D ticarcillin. So which of the following class of penicillins is synthetically formed, acid stable, and can be taken orally? Penicillin G, penicillin V, ampicillin, ticarcillin. Which of the following class of penicillins are synthetically formed, acid stable, and can be taken orally? So there are three different criteria are given. First criteria is that it must be synthetically formed. Second is it must be acid stable, and the third one that it can be taken. Can be taken orally. So which of the following class of penicillin fits into these three criteria? Okay, let me check the chat box. I have one answer which says it is number D. Let me check if number D is the correct answer or not. Okay, so correct answer is actually number C, ampicillin. Okay. So the classification of penicillins is another very important aspect. So first is the source. So what are the different source? It can be natural. For example, is the penicillin G. It can be the biosynthetic. The example are penicillin V, penicillin F, penicillin K. And we have the another option which is the synthetic, which is ampicillin, amoxicillin. Then the spectrum of activity, it can be narrow, which is the for methicillin, oxacillin, and nephcillin. The spectrum of activity can be broad for ampicillin and amoxicillin, and it can be extended like for ticarcillin and azocillin. Then we have the penicillin, the resistance to acid, which is if it is acid stable, for example, ampicillin, penicillin V, cloxacillin and oxacillin. So these are acid stable. Then acid unstable are the ticharcillin, methicillin, piperacillin and penicillin G. So these are the acid unstable. Now the route of administration, it can be oral like ampicillin, amoxicillin, cloxacillin and dicloxacillin. These are the oral route of administration, and parenteral. These are the penicillin G, methicillin and ticharcillin. Okay, so the penicillins which is synthetically formed, acid stable and can be taken orally is the amoxicillin. In the all the options is ampicillin is the one because it is the synthetic. Okay, synthetically formed. It has a broad spectrum of activity. It is acid stable and also it can be taken orally. Okay, so number C, ampicillin was the correct answer. Now, next question. Which of the following statements is false regarding the vancomycin? Your options are number A, it inhibits the transglycosylation and transpeptidation. Number B, it is a broad spectrum bacterial antibiotic. Number C, it is the mostly prescribed to treat Staphylococcus aureus infection. And number D, none of those. None of these. Which of the following statements is false regarding vancomycin? Again, I will say number A, it inhibits the transglycosylation and transpeptidation. Number B, it is broad spectrum bacterial antibiotic. Number C, it is mostly prescribed to treat the Staphylococcus aureus infection. Number D, none of these. Identify the false statement regarding vancomycin. Which of the statement is false regarding vancomycin? Let me check the chat box now. I have one answer which says it is number B. Okay, let me check. Yes, number B, it is the correct answer. The statement which is false regarding vancomycin is number B, which says it is a broad spectrum bacterial antibiotic. So actually, vancomycin is a narrow spectrum bactericidal antibiotic, and its mode of action is like it inhibits the glycosylation and transpeptidation. The therapeutic uses is the MRSA infection. Vancomycin caps the pentapeptide tail by hydrogen bonding and acts as a steric shield, blocks the access of the transglycolase or transpeptidase. It is a narrow spectrum antibiotic, but it has a bactericidal effect. That means it is going to kill the bacterium. So it has a unique mode of action like penicillin. Vancomycin is also going to inhibit the transpeptidase enzyme, but along with that it also inhibits the transglycosylase enzyme. Because of this macromolecular structure, vancomycin caps the pentapeptide tail by hydrogen bonding and thus acts as a steric shield, blocking the access of this pentapeptide to membrane bound enzymes such as the transglycolases or transpeptidase. Okay. So the statement number B, which says that it is a broad spectrum bacterial antibiotic, this is the false because it is a narrow spectrum. Now problem number 19, it says that which of the following statements regarding Pseudomonas aeruginosa is false? Your options are number A, this is an example of opportunistic pathogen. Number B, the cells of the P. aeruginosa are cone shaped. Number C, it prefers to grow in moist environment. Number D, it has intrinsic resistance to a wide variety of antibiotic. Which of the following statement regarding Pseudomonas aeruginosa is false? Number A, it is an example of the opportunistic pathogen. Number B, the cells of P. aeruginosa are cone-shaped. Number C, it prefers to grow in moist environment. And number D, it has an intrinsic resistance to a wide variety of antibiotics. Which of the following statements regarding Pseudomonas aeruginosa is false? Let me check the chat box. Okay, number C is the given answer. So the statement which is false regarding vancomycin is number B. It is a broad spectrum bacterial antibiotic. Okay. So glycopeptide. Okay. So this one is okay. This one. The statement regarding Pseudomonas aeruginosa is false. It is number B. The cells of P. aeruginosa are cone-shaped. This statement is false. So Pseudomonas aeruginosa. This is the reactions which is due to because of the infection of Pseudomonas aeruginosa is the bladder catheter infection, urinary tract infection, skin-borne infection, implant associated infection, osteomyelitis, pneumonia, and catheter related infection. So Pseudomonas aeruginosa is an example of an opportunistic pathogen. Such as organisms are not normally harmful to healthy individual. Indeed, many people carry this organism without being aware of it because their immune system keeps it under control. Once that immune system is weakened though, the organism can start multiplying and lead to serious illness. Hospital-bound patients are particularly at risk, especially those suffering from a shock and AIDS, or those undergoing cancer chemotherapy. Burn victims are particularly prone to P. aeruginosa skin infection, and this can lead to septicemia, which can prove fatal. The organism is also responsible for serious lung infection among patients undergoing mechanical ventilation. The cells of the P. aeruginosa are rod-shaped and can appear the blue and green in color, which is why it was given the name aeruginosa. It prefers to grow in moist environment and can be isolated from soil, water, plant, animals, and humans. It can even grow in distilled water and contact lens solutions in hospitals. There are several possible sources of infection, including respiratory equipment, sinks, uncooked vegetable, and flowers by the visitor. Pseudomonas aeruginosa is a difficult organism to treat because it has an intrinsic resistance to a wide variety of antibacterial agents, including many penicillin, cephalosporin, tetracycline, quinolones, chloramphenicol. There are two reasons for this. The outer membrane of the cell has a low permeability to drugs. Even if the drug does enter the cell, there are efflux system which can pump it back out again. Nevertheless, there are drugs which are proved effective against this organism, in particular aminoglycosides such as the tobrasmycin and gentamicin. The penicillins such as triclosan are this often given in combination of each other. Okay, so number B was the correct. Now, last question of today's session is says which of the following is not a fourth generation cephalosporin? Your option are cephalexin, which is A, then cefoxyticin, which is B, ceftazidimine, which is C, and cefpirimorum, which is D. Your options are number A, both A and B, number B, A, B, D, number C, A, B, C, and number D, C and D. Which of the following is not a fourth generation cephalosporin? Okay, let me check the chat box. I have one answer which says it is number A. Let me check if number A is the correct answer or not. So the correct answer is, it is number C. A, B, and C. These three are not fourth generation cephalosporin. So this is the structure of fourth generation cephalosporin. So in general, cephalosporins are okay. Let me on the laser pointer. In general, cephalosporins are useful broad spectrum antibacterial agents for the treatment of the septicemia, pneumonia, meningitis, biliary tract infection, peritonitis, and urinary tract infection. Cephalosporin C itself has been used in the treatment of urinary tract infection as it is found to concentrate in the urine and survive body's hydrolytic enzymes. First generation cephalosporins have a good activity against gram-positive cocci, and they can be used to treat some community-derived gram-negative infections that are caught in hospital. They can be used against the S. aureus and streptococcal infection when the penicillins have been avoided. Cephalotin is the active than penicillin G against some gram-negative bacteria and is less likely to cause allergic reaction. It can also be used against the beta-lactamase producing S. aureus strain. Cephalexin is useful for the treatment of urinary tract infection which do not respond to other drugs on which occur in pregnancy. It is also useful in treating infections of the respiratory tract, ear, skin, and mouth. Cephalexin is recommended for the use as the prophylactic prevent infection when surgical procedure are used to implant the foreign bodies. Second generation cephalosporin in general, the second generation cephalosporins have variable activity against gram-positive cocci but increased activity against gram-negative bacteria. Cephalosporin is active against the flora, including Bacteria frangilis, and was once recommended for the peritonitis. Cephalosporin was the wide spectrum activity and use against organisms which have become resistant to penicillin. However, it is not active against difficult bacteria such as P. aeruginosa. It is used clinically against gonorrhea and respiratory infections, plus influenza, and so on. It is also used in surgical prophylaxis as well as the treatment of Lyme disease. Cephalosporin is used in surgical for the treatment of gonorrhea. Third generation cephalosporin have good activity against gram-negative bacteria but vary in their activity against gram-positive cocci. The ability to attack the organosa also varies from the structure to structure. They lack the activity against the MRSA organism and Enterobacteriosis. Cephalosporin is an injectable cephalosporin which have excellent activity against P. aeruginosa as well as other gram-negative bacteria because the drug can cross the blood-brain barrier. It can be used to treat meningitis compared with the other aminothiazole structures. Cephalosporin has a good activity against staphylococci but loses activity against strain of the methicillin susceptible S. aureus. It is because the decreased binding affinity for the transpeptidase enzyme present in the S. aureus or cephalosporin is used to surgical prophylaxis as in cephaloactive meningitis. Fourth and fifth generation cephalosporin, fourth generation cephalosporin have activity against gram-positive cocci, including P. aeruginosa and many of the Enterobacteria species. Cephalosporin is administered as an intravenous injection and infusion that has been against a variety of sensitive and gram-positive, gram-negative bacteria. Phosphomase is used to licensed for the treatment of bacterial pneumonia and acute bacterial skin infection. So as you have seen that cephalosporin is the fourth generation cephalosporin. So A, B, C is not the fourth generation. So if option number C is the correct one. Okay. So this was the last question of today's session. So today we discussed mainly about different antibacterials, especially the penicillin. Okay, so now I will show you where to find this particular problem solving session. You have to go to the SWAM portal first, to the course web page of the organic chemistry and biology and drug development. If you log into it, once you log into the SWAM portal, what you will find, I will show you. So here is the course content and announcement section. Here you will go to the problem solving session. If you go to the recording, you will be redirected to a page. If you click here, you will find here all the YouTube links of the classes and Google Drive folder where I upload the course material. Okay, so you can go to that portion and you can find it. So thank you for joining today's class. I will close the session for today. For any problem, any query, I will share my email ID. Here you can, okay. In this email ID, you can drop your query or doubts, which I will feel happy to answer. So today it was our week 9 live session. Now we will meet on the next Monday from this time 6 to 8 p.m. and we will discuss the next week's course content. Okay. Thank you all for joining. I will close the session for today.